Good afternoon, dear students. Myself, Dr. Adarshi Bayan from Arya Vidyapeeth College, Guwahati, and today I am going to deliver an online lecture on organic spectroscopy. In today's lecture, I will be specifically covering about some important terminologies used in UV visible spectroscopy. So let us proceed. So the very first important terminology used in UV visible spectroscopy is the term chromophore. A chromophore is defined as an isolated covalently bonded group that shows a characteristic absorption in the UV visible region. So basically, chromophore is an atom or a group of atoms present in an organic molecule that is responsible for imparting color to the particular compound. For example, azobenzene is an organic compound which has red-orange color due to the presence of the chromophore azo group. Similarly, nitrobenzene is another organic compound which is pale yellow in color due to the presence of the chromophore nitro group. The second very important terminology in UV visible spectroscopy is the term oxochrome. So basically, oxochrome is a substituent which is covalently attached to a chromophore and which is responsible for enhancing the color of the particular organic compound. For example, the amino group. When this amino group is attached to this particular azobenzene, what it does is it enhances the color of the particular compound. Similarly, when this amino group is attached to this nitrobenzene, it enhances the color of the nitrobenzene. So here the amino group is acting as a oxochrome which attached to the chromophore enhances the color of the particular compound. The next important terminology used in UV visible spectroscopy is the bathochromic shift. So the bathochromic shift is the shifting of the absorption maxima peak towards longer wavelength or lower energy. This particular shift is sometimes also referred to as a red shift. This is due to the fact that the longer wavelength, the color represents the red color in the visible spectrum. So therefore, the shifting towards the longer wavelength of a particular color of an organic compound is called red shift. For example, benzene. Benzene, which has a absorption maxima around 256 nanometer, undergoes a red shift when a oxochrome amino group is attached to the benzene ring. So when an amino group is attached to the benzene ring, the absorption maxima peak shifts to 280 nanometer. So from 256 it red shifts or undergoes bathochromic shift to 280 nanometer. The next important terminology is the hypsochromic shift. The hypsochromic shift is the shifting of the absorption maxima peak towards shorter wavelength or higher energy. This particular shift is also called blue shift because the shift towards the shorter wavelength represents the blue color. So that is why this shift is also called the blue shift. For example, aniline, which has an absorption maxima of 280 nanometer, undergoes a blue shift when it is converted to anilium chloride. So here the oxochrome undergoes a conversion to form an ionic compound. So as soon as it forms an ionic compound, the absorption maxima reduces to 265 nanometer. So here aniline undergoes blue shift. The subsequent important terminology in UV visible spectroscopy is the hyperchromic shift. So the hyperchromic shift is an effect that results in the increased absorption intensity of a absorption peak. This particular shift is usually observed due to the introduction of an oxochrome in the structure of an organic compound. For example, pyridine, which has an intensity of 2750 in the UV visible spectrum undergoes a hyperchromic shift when the oxochrome methyl group is attached to the pyridine ring. So as a result, the intensity of the peak undergoes increase. So as you can see here, the peak undergoes increase in intensity upon the addition of the oxochrome methylene group in the structure of pyridine. So here we say that pyridine undergoes a hyperchromic shift upon introduction of the oxochrome methyl group. And finally, the last important terminology used in UV visible spectroscopy is the hypochromic shift. So the hypochromic shift is an effect that results in decreased absorption intensity. So this type of shift is usually caused by a group which distorts the geometry of a particular molecule. For example, naphthalene. So naphthalene is a planar organic compound which shows an intensity of 19,000 in a UV visible spectrum. Upon addition of the oxochrome methyl group in the structure of naphthalene, the structure of naphthalene undergoes distortion. So due to this, the intensity of the naphthalene reduces to 10250. So this decrease in intensity is hypochromic shift, and it is due to the introduction of the oxochrome in the structure of naphthalene which distorts the structure. So students, these are some important terminologies that are used in UV visible spectroscopy quite frequently, that is the chromophore, the oxochrome, the bathochromic shift or red shift, the hypsochromic shift or blue shift, the hyperchromic shift and the hypochromic shift. Thank you.